Trying to get my chair all situated. Sorry. Okay, so cell membrane structure. It's really important that we have the cell membrane because it keeps things where it's supposed to be and it makes us not break. So it's kind of important. So the phospholipid bilayer with embedded proteins, this is the fluid mosaic model. And this functions in providing a site for energy reactions, nutrient processing, and synthesis of various modalities in the cell. Fancy word, modalities, right? Huh. Okay, so the passage of nutrients into the cell and discharged waste also occurs, and then the cell membrane is selectively permeable. All right, so what does it look like? Well, it's pretty complicated, right? But it's actually really cool. So the phospholipid bilayer, there's two things that you can think of. It's made of phospholipids, and the bilayer literally means that there's two layers of phospholipids, right? Here's one layer and there's the other layer. They're hydrophobic tails that are scared of water, so they're like kind of hidden by the polar heads, right? But also, when you look at a phospholipid, it has two tails. Okay, so if it asks you to identify things, look for the phosphate head with two tails, so you can use the bilayer as a trigger word for both of those. But you have transport channels, proteins, glycolipids, phospholipids, the cell glue, which is the cytoplasm, etc. All right, inside the bacteria cell, in the cell cytoplasm, this is, like I said, kind of goo. And this is dense gelatinous solution of sugars, amino acids, and salts. And it's about 70 to 80% water. This is why it's important that we keep hydrated, right? And this serves as a solvent for materials that are used in cell functions. Looking at the nucleoid, this is where our chromosomes are, right? So this is a single circular double-stranded DNA molecule that contains all of the genetic information that's required by a cell. We have plasmids, and these are free, small, circular, double-stranded DNA pockets. And these are not essential for bacterial growth and metabolism, but they are used in genetic engineering, and these are readily manipulated and transferred from cell to cell. And these are those little pockets that you can see. So moving on with like the different types of organelles, we have the bacterial ribosomes. And ribosomes are made up of 60% ribosomal RNA and 40% protein. And these consist of two subunits, a large and a small subunit. Okay, and then the prokaryotic cells differ from eukaryotic ribosomes in the size as well as the number of proteins. Also, the site of protein synthesis, and these ribosomes are found in all cells. Here is an example of the ribosomes. You have a 70S, a large subunit is a 50S, and then a small subunit is the 30S. And no, these don't add up to 70. I don't know, remember why they named each of them the way that they did, but they are. So there's the ribosome, which is a 70S, the large subunit, which is 50S, and then the small subunit, which is the 30S. And moving on to bacterial internal structures, we have inclusions and granules. And these are intracellular storage bodies, and they vary in size, number, and content. And the bacterial cell can use them when the environmental sources are depleted. And so here is a fun little picture of some of the internal structures in the bacterium. Okay. And here's another one that you can see the internal structures. The cytoskeleton, this is like a skeleton, right? It's a structural thing. And so many bacteria possess an internal network of protein polymers that are closely associated within the cell walls. And so you can see when they're dyed green here, these are the actin filaments that are big proponents of cell structure. We also can have endospores, and these are inert, resting cells that are produced by some gram-positive genera, such as Clostridium, Bacillus, and Spirosaccharina. And these have two phases of the life cycle. The first is the vegetative state, and this is where it's metabolically active and growing. And then we have an endospore, and this is kind of like the storage, right? Where it can be exposed to adverse environmental conditions, capable of high resistance for very long-term survival. There's some story, and it's like somebody found some old like canning jar down in their basement, And I don't know why they decided to open up. All these spores were released, and it ended up like starting not the bubonic plague, but something kind of bad, because these endospores were just chillaxing, and then when they were opened, it got open to an environment that was conducive for its growth, and it ended up reconstituting and growing again. Okay. You also have sporulation, and these would be a formation of endospores, and these are the hardiest of all forms, and these can withstand extreme heat, drying, freezing, radiation, as well as chemicals, and these are not a means of reproduction, but like I just said in the kind of story, when you germinate it, then it can return to that growth stage. So here is a sporulation cycle where you go from your vegetative state all the way hibernation, and then during germination where you can um, continue to grow again. Okay. So endospores then are going to be dehydrated, metabolically inactive. They have a thick coat. The longevity verges on immortality. They're very hard to go away. There are some that we found that are 250 million years old. They are resistant to ordinary cleaning methods and boiling. And in order to kill them, we have to pressure steam them at 120 degrees Celsius for 20 to 30 minutes. And that's the only way that we can destroy them. So they're very, very hardy. So here is a little check for you. It already tells you the answer. So if you discovered that the bacterium you're studying stores phosphate, which of the following would you expect to contain the stored phosphate? And the answer would be granules. Alrighty.